they're just being redistributed toward the shaded side. So let's see how we got that. First of all, scientists were observing that the same amount of auxin was being found in coleoptile tips in both the dark and the light. So it doesn't, that sort of speaks against auxin being destroyed by light, right? Sort of dismissing that alternative hypothesis. They also found that following directional light exposure, more auxin was found on the shaded side. So that speaks positively for the redistribution hypothesis, that light is driving auxin towards the shaded side. What they also found is that if they put a thin piece of glass through the shoot tip and then lighted it from one side, that differential distribution of auxins disappeared. So if you block the auxin's ability to travel to the shaded side, you get the same amount of auxin on the shaded and the light side. So all of that together speaks against the auxin destruction hypothesis and speaks in favor of the auxin redistribution hypothesis. So it seems then that sunlight is driving auxin towards the shaded side, and therefore only the shaded side has cell elongation, which causes the bending towards the light. And this is just some pictures of some of those experiments. Okay, but how does the plant know? I mean, plants, they don't really know, that's why I've got it in quotes, but how do they detect where the light is? How are they picking up on this? We're going to talk more about this later on, but it seems like this redistribution of auxins in these shoot tips is mediated by what are called photoreceptors. So plants have photoreceptors, or light sensors. These are pigment-containing proteins that absorb sunlight and convert that signal into a biochemical response. It's really an amazing thing. So they have these photoreceptors. This phototropic response that I just told you about, this bending of shoot tips towards the sunlight, is triggered by wavelengths of blue light, particularly. So these photoreceptors seem to be responding to this 400 to 500 nanometer light. And so because of that, we think that these photoreceptor proteins are related to other well-studied proteins that bind pigments called flavins and that absorb blue light. The reason why I'm telling you that is we don't know a whole lot yet about these photoreceptors. But because they seem to be absorbing blue light selectively absorbing blue light, it seems like they might be working sort of like these other well-studied proteins that bind pigments, bind flavins, and absorb blue light. So anyway, that's where the research is kind of headed. Okay, so that's a little bit about phototropism. Gravitropism. Plants grow in response to gravity. And like I said before, shoots are negatively gravitropic. Now, that one's a little bit hard to tease out because if you think about it, shoots are probably more positively phototropic than they are negatively gravitropic. Right? That light is plenty of a stimulus to get them to grow in the right direction. But it's probably really easy to buy into the notion that roots are positively gravitropic. A root can't see the sunlight, and so roots are orienting their downward growth by growing with the force of gravity. This is really amazing. But if you put a plant on its side, right, lay a plant down on its side, the auxins are going to be naturally redistributed to the lower side of that stem or root. You know, stems and roots. I'll show you a picture like this here. Okay, laying a plant on its side. The auxins are automatically, naturally, by way of gravity, going to be redistributed to the lower side of that stem and the lower side of that root. Now, watch how they react. They react oppositely. In the shoot, the higher auxin concentration on the lower side stimulates rapid cell expansion on that bottom side, which causes the stem to curve back upwards. All right, look right here. All the auxins are being redistributed to the bottom side of this stem. That causes cell elongation on the bottom, not on the top, on the bottom, which causes it to bend back upward and to get back into the light. A plant cannot afford to be laying on its side. Right? It needs to grow up towards the sun. We can't see this, but the roots are doing the exact opposite inside the pot. Those roots are curving back down that way. Okay, we can't see it, but that's what's happening inside the pot, and that's because they're reacting in an opposite fashion to what's happening in the roots. Look, the auxins are still sinking to the bottom side of those roots, but roots are more sensitive to auxin, right? And so the increased auxin concentration on the lower side of the roots inhibits cell expansion on the bottom side. Right? The top side is still expanding normally, and so then you get a downward bending in the root because of the inhibition of normal cell expansion on the bottom side of the root. Okay? And here's a great example. Here are two parts of the same plant responding in the exact opposite direction to the same auxin stimulus. And it's because roots have an increased sensitivity to auxin. And so the auxin inhibits their cell expansion, where in the shoots it leads to increased cell expansion. So the target tissue matters in this case. Okay. Has anyone ever grown upside down tomatoes? I haven't seen that lately. That was sort of a big craze 3, 4, 5 years ago. You probably can still go to Lowe's in the spring and get a upside down tomato kit. I did that one. Or they call them hanging tomato plants.
Topsy-turvy something. Yeah, it's top.
Yeah, that's right. So the tomatoes kind of, your tomatoes are going to growing down, but they're still curving up towards the light. Like this one. All right, look. This should be no surprise to us after the last lecture. Recent studies have shown that calcium plays an important role in these gravitropic responses of both shoots and roots. All right? And that the response is mediated by a calcium binding protein, right? A protein kinase called calmodulin. Okay? So check this out. As the auxins are moving to the bottom side of the shoot, the calcium ions are moving towards the upper surface of the shoot. All right, before it curves upwards. And while the auxins are moving towards the lower surface of the root, the calcium ions are also moving towards the lower surface of that root before it curves downwards. So we don't know exactly what's happening after that. We do know that the calcium ions are binding to a protein kinase, and that, we would guess then that those protein kinases are phosphorylating some other protein, and there's probably something else downstream to cause that final response, that bending downward, that bending upward. So it's not as simple, right, as just, here's the hormone. There's going to be that hormone response pathway following that in most cases, and it's not surprising that calcium ion are a couple steps down that pathway. That's not surprising. All right, but how do plants know which way is up and which way is down? Well, there are gravity. They have gravity perception sites. They have gravity sensors, and these gravity sensors are called ameloplasts. This is my favorite part, maybe of the whole semester. I don't know why. It's really cool. I really get it. These ameloplasts are little starch-containing plastids, right? Little starch-containing organelles. Right? A chloroplast is a plastid. Plastids kind of store stuff in plants. So these little ameloplasts, these starch-containing plastids, and they're found in specific cells of the shoot and the root. Now in the shoots, they're found in the region of the vascular cambium. In roots, they're found in the columella, the central column of the root cap. And I want to kind of follow that root model here. That's what our pictures show us. All right, so look, here is the root cap, the very end of the root, right? These central cells are called the columella cells. You see those little dark starch grains? Those are the ameloplasts. Those are the gravity sensors. All right, here they are. Look, this is amazing. Here's one of those central columella cells. Look at these. Here are the ameloplasts here. Now look, I want you to notice, I want you to tell me something. This root is oriented like this. Where are the ameloplasts?
On the bottom.
They're on the bottom. Of course they're on the bottom. They settle towards the bottom. All right?
No, there's no matter the orientation.
Well, no matter the orientation, and I'll show you that in a second. If it's oriented like this, they're on the bottom. What if I knock the cell over? Where are they going to be? They're going to roll over to the side. If I turned it upside down, they're going to be over here, which what used to be the top. And the cell can detect where they are because they're pressing on the cell membrane. And the cell can feel that. Right? So the way these things are, wherever they're settling, the cell can feel that, and that's how it knows which way is up and which way is down. Because the plant knows, quote unquote, that this is the bottom side of the cell. And it knows that this is the top side of the cell. And it knows that this is the side. And so if these things are resting on the bottom, then everything's okay.
Oh, see, I thought even like you said, the root is down, and like even if it is oriented up, like it would still be considered the bottom, like.
No.
So.
This is always the bottom. If we flip it, now the bottom is up. And the cell knows those things will settle to what used to be the top, and it'll be like, that's not right. I got to make this right. And so it'll grow so that the bottom's down again, and that those things are settle out on the bottom. Isn't that amazing? So those little ameloplasts, it's really the sedimentation of those ameloplasts, or the settling of the ameloplasts in conjunction to what the cell can feel, that's where gravity perception is bound up in that. So it may not be exactly right to call the ameloplasts themselves the gravity sensors. It's the interaction of those ameloplasts with the cell membrane in most cases that lets the plant know which way is up and which way is down. Okay, so if I were to just sort of be explicit about this, I don't need to be explicit about this. It's just words getting in the way of what I've already said. All right. So we call these ameloplasts, they're called statoliths, or gravity sensors. The book actually does call them gravity sensors. All right, so let's look at a picture. And these words here just narrate what I've already said. It narrates the picture too. All right, so here's the cartoony version. Look, here they are. This is the bottom. This is supposed to be the bottom. That's the top. These are the sides. And look, they're all settled down on the bottom. And even the cell above it, they're settled on the bottom. You knock that cell over like this, boom. All of those things roll to the side. And then it's like, no, this is not right. These little balls are not supposed to be on my side, they're supposed to be on my bottom. Right? And so the plant will reorient its growth, and it'll grow, it'll grow downward so that those little balls reorient to the bottom. Yeah?
How fast do those things move throughout the tip to the bottom?
This whole thing is on the order of several hours, so it's not an instant thing, right? I mean, the curving, well, they probably move pretty quickly.
Yeah.
It may not be like boom and they're down, but they roll pretty quick. But to get the overall response of the root growing downward again takes a few hours. I like to call these little beach balls. They look kind of like beach balls. If you stretch it, right? So now, if you stretch it, they look like beach balls. See, John? So that whole thing I described is called the starch statolith hypothesis. The way that these little starch grains, these plastids, are behaving like statoliths or gravity sensors. I really like that a lot. There's a couple of other alternative hypotheses. The hydrostatic pressure hypothesis says that plant cells detect gravity changes by sensing hydrostatic pressure exerted by the protoplast on its cell wall. So what that means is